Good morning, good afternoon, good evening, good night from wherever you're joining us today, and welcome. So I am Liona Moldovanova, and I manage site investigation supply. We are a small company based in Sydney and Queensland, and we manufacture and supply consumables for environmental and geotechnical site investigations. We are also the exclusive distributor of Microbial Insights in Australia, and we are very proud and happy to bring these new technologies in the country. And we have today Dora Taggart. She is the CEO of Microbial Insights. If there is anyone in this world who knows about this topic, that is Dora. So thank you so much, Dora. We will have Q&A at the end of the webinar. There's a little hand on top of your screen which says raise. Just feel free to click on it if you have any questions, and we will unmute you so you can ask Dora whatever you need to ask. And yeah, so again, thank you for being here today, and thank you so much to Legion Drilling, Jonas, for helping us organizing this webinar. If you have anything to say, it's your time.
And I just thank you, Elena, for helping to organize this. Thank you, Dora, of course. Thank you, everybody, for attending. The only thing I would like to mention is like I know Dora mostly from collaborating in the preparation of a book on contaminated land and remediation that we published last year. It was a pleasure working with her. And this webinar will be a bit more focused on mining applications, but as many of you probably know, they can be used for many other things like remediation or bioremediation of petroleum hydrocarbons, chlorinated solvents, monitor natural attenuation, and so on. But it's great to see how this is growing as well in the mining sector, and I'm really looking forward to the presentation. So Dora, all yours.
All right, thank you so much for both of you for organizing this and setting it up for us, and for the kind words. And thank you to everyone that's taking time to be here on this webinar with me today, either live or the people that are going to watch the recording. We've had quite a bit of interest around the world because I think it's definitely a global topic that we should be talking about. So I'm really excited to present my view of what we're currently doing and kind of what the future holds. And then I would love to talk to you guys more about it, whether it's on this platform or if you want to reach out to me afterward to talk to me about what you guys are doing. I would love to learn more from all of you today. So let me go ahead and start my slides here. So my goal of this talk today is to show you why I think microbes are really important in mining, and we've learned a lot about applications in the subsurface and how to do good analysis from environmental samples from the past 25 years of bioremediation. And so I've been doing this for a long time and have gotten to see a lot of sites and a lot of technologies around the world, and I'm excited to share with you my passion for these microbes and what we're trying to do. And then I want to give you a couple of tools that we use commonly that I think are really important for the mining industry, and show you 2 case studies where we've used the tools. And then for those of you that are already experts in this, if you'll bear with me at the end, I'm going to show you something I think is brand new for most of the industry. And we're going to talk about some new technology that is going to be released very soon that I think has a lot of promise. So that's kind of the view of what we're going to be doing today. So I wanted to start by sharing my passion for microbes. Understanding their role in the processes involved in the subsurface has really been my life's work for the past 25 years. And I think microbes are just the key foundational part of a lot of processes that we're interested in. And if we can understand the benefits of these microbes and start to understand their functions in a variety of environmental processes, particularly in mining, we can really harness the power of these microbes so that we can use them to help us either mobilize metals that we're interested in. So sometimes we might want to increase movement through certain parts of a process, Or other times, maybe we want to immobilize certain metals and precipitate them out of solution at other parts. And microbes are really key to being able to do this. So we can look at this across a variety of locations and really harness the energy of what these microbes have. But one of the things we used to worry about is, can microbes actually survive in these extreme mining environments? And so we used to do things like plate counts or bug bottles. And many of you on the call, I've saw some of the names. I know you're familiar with those processes. And what we found is less than 1% of microbes like to grow in a bug bottle or on a plate in the lab. We just don't do a good job of giving them what they need to survive, especially replicating these extreme environments like we have in mining. So we can't give them, the right pH or the right temperature, all these things that can be important. So we used to think they didn't survive and they weren't present in these environments. But with the use of molecular tools like we have today, we're actually able to understand that not only are microbes present in these extreme environments, but they actually, some of them really thrive under these conditions. So organisms like acetophiles can actually thrive in really low pH of 2 to 3, or sometimes even lower. There's an organism I'm going to show you today that was surviving at a 1.5 pH. And then there are certain groups of organisms that really are not bothered by high metal toxicity like other organisms are. So they can help us with these high metal concentrations that are present in different locations. And a group of organisms that we really need to become familiar with in mining are called chemolithotrophs. And these are organisms that gain their energy either by oxidizing or reducing inorganic compounds. And as we know in the mining environment, these are extremely rich in metals and in minerals like iron sulfides and other elements that actually make a great home for these organisms to be able to live and to thrive. So in these environments, we'll see organisms like Acidothiobacillus ferroxidans. And this organism in particular can not only live and thrive in the mine, but it can help us extract metals through a process called bioleaching that we'll talk about today in this presentation. There's also organisms like Leptospirillum and Thiobacillus that sometimes might be a cause for concern, as they're key players in the formation of acid mine drainage because they can oxidize iron and sulfur into sulfide minerals that will eventually result in sulfuric acid. But if they're present in certain parts of the process, they may actually be helpful to us because they can actually be utilized to help extract metal from our ores. So this is why we really need to be able to use molecular biological tools so we can harness the power that these little tiny microbial powerhouses have. And we can use this appropriately to help us optimize our processes, to maximize metal recovery. We can mitigate environmental concerns, just like we've been doing in bioremediation for other processes. And we can use it to remediate toxic compounds that are generated during these processes. But in order to do this, we need to have direct analytical methods that don't require the cultivation like we used to do. And that's where these molecular tools really come into play. So those of you that know us know for the past 25 years, we've been using these tools in environmental remediation processes, particularly qPCR that we'll talk about. Even earlier, we were doing lipid analysis, looking at ways that we could manage environmental remediation for things like explosives. We did a lot of work in uranium sites, a lot of high nitrite and nitrate levels, looking at movement of heavy metals. So since the early 90s in particular, these tools have been commercially utilized in remediation type processes. And they actually are very powerful for use in the mining industry as well. So if you think about something that can be both a liability for us and also can cause complications when we get to a closure standpoint, things like acid mine drainage will come into play. And that's certainly almost always a concern. So when we perform mining activities, as many of you know, the buried rocks are exposed to air and water, and this allows those sulfur-bearing minerals to be exposed and to react, producing sulfuric acid, which lowers the pH of our water in the process. And then sometimes we have compounds like pyrite that might be present, and pyrite oxidation can actually speed up acidic production even more. So then as that pH keeps lowering and dropping, metals are leached from the surrounding ore and the rock formation. And while that's great if we want to do recovery, if those aren't collected, that acidic water is now also filled with metals that can be hazardous to nearby receptors like lakes, rivers, and streams. So it's definitely something we want to think about. And the earlier we think about managing this process and controlling what's happening in our systems, the better it's going to be for mitigation. So if accidents happen along the way, we have less of this accumulation to deal with. And also, rather than just keep postponing, we can start to deal with the problem a lot earlier so that when we get to closure, it's not such a daunting task. And if we recover some of the metals even better along the way, then that's really useful to the process of what we're trying to do with mining. And we can actually use this costly situation to help us turn into something that can have an economic benefit as well. So there's a lot of research that's being done on this particular topic with some great university work around the world. There's a lot of national labs in the US that are working on this and have been working on this. And so we've learned a lot about these different processes. And by learning about these microbes, we can start to understand what they're doing so we can then maximize these uses. So if we think about the microbes that are involved, two of the key groups that we think about for oxidation are sulfur oxidizing bacteria and metal oxidizing bacteria. And these are important because sulfur oxidizers produce sulfuric acid, and metal oxidizers can actually oxidize the dissolved metals, leaving behind heavy metals or rust in different situations. And this can really affect our process. We can get everything from buildup of metals in certain situations. You can get increased scaling that can happen in systems, biofilm formation. Plus, while these aerobic organisms are utilizing all the oxygen that's available to them and doing these processes, we're actually creating more reducing conditions. So what may start in one part of the system as an aerobic process that's occurring can quickly shift to where our community or our village of microorganisms have changed. And so these reduced conditions give us growth for other organisms that like to grow and thrive in those anaerobic conditions. We can get things like methanogens and fermenters, acetogens, and other metal oxidizing or reducing bacteria that are present. And this will change what happens in the cycling of the compounds. And so the process itself changes, the redox conditions change, the microbes that are present change, and now we have new compounds that are being generated. Like in this case, maybe we start to produce a lot more methane in these situations. And then you have acidophilic methanotrophs that actually thrive in that condition. And so then they start to oxidize the methane. So we need to really understand these processes and how they interact so that we can use them to our advantage. And not all organisms are doing something bad in our system. Microbes, just like in our the human health, there are bad microbes that can cause problems and cause infections and diseases. There are also good microbes, like in our gut microbiome, that help us with digestion. The same thing happens in mining and in remediation, where we can use these processes for the good of what's happening. So in situations like this, we can look at sulfate reducing bacteria who will produce sulfides that can help us precipitate dissolved metals. So we can use the SRBs not only for the change that's happening in the sulfide formation, but we can also use it to precipitate the metals that we want at different parts in the process. And then there's organisms like arsenite oxidizing bacteria, and they'll take arsenite and change it to a less toxic arsenate. So if you have that in your system, looking for these organisms can be very valuable. And then there's jarosite producing microorganisms that can also help with bioremoval of arsenic. They actually adsorb this compound and this harmful metalloid into the microbial cell. And we'll talk about that just more in a minute as we look at different processes that microbes can do. And thinking about that, one of the important things we are talking about on a global basis is how do we do a better job of recovering rare earth elements? Every country around the world is thinking about this because they're crucial for modern technology. We've really had a huge uptake in the use of these metals over time, from everything in our cell phones or the laptops we're on right now to thinking about the future and green energy production. I was just at a conference in Nashville last week thinking about how energy is changing and moving from oil and gas to new energy. And with some of this green energy production, we're going to need more rare earth elements. So their unique properties like magnetism and luminescence are going to be very important for us on a global scale. And we call them rare earth elements, but actually these compounds are pretty abundant in the subsurface. The problem is they're rarely found in concentrations that are economically viable mineral forms. And so it can make recovery and transformation of these a really important concern for us to address. So how do we improve the recovery of these critical metals? Well, first, we have to understand how these microbial mechanisms can be beneficial to us. And there's three main ways that I thought of when we were putting this presentation together. The first is biosorption, where we can use biological materials like bacteria, algae, or fungi to selectively bind and remove them from various sources like industrial waste or wastewater. And this occurs through mechanisms like adsorption, absorption, and ion exchange. And when you think about biosorption, this is very cost effective. These organisms are naturally present. They naturally have this capability for biosorption to happen. And so it's a very environmentally friendly alternative to some of the traditional, really invasive methods of recovery that we use today. So we can use less solvents, we can have less manpower that's involved in this if we harness the power of the microbes and start to truly use those better. Another tool we can use that microbes possess is something called bioaccumulation, and I mentioned this with arsenic just a minute ago. There are microbes that will naturally produce organic molecules that change the oxidative state of metals, or they chelate them, and that makes them bioavailable to the cell. So as this happens, the cell might actually uptake some of those metals and retain it inside their cell walls or in the cell wall itself. And this happens differently for different organisms and to varying degrees. But through great research, we're starting to learn which compounds that they can chelate and utilize and which ones are kind of held within the cell. So that can be beneficial. And when we get to the end of the presentation today, I'm going to show you some of the future of how I think we can use this. So finally, we have the ability to utilize bioleaching to help us for recovery. So as microbes release these organic compounds and the metals solubilize, the rare earth elements and other metals can actually be released into solution. So then processes like precipitation or solvent extractions or ion exchange can be done to recover them better from solutions. And you can get a lot better recovery rates than you can without the bioleaching being involved. So again, this makes it a more environmentally friendly, energy efficient, and cost-effective part of the process. It's also really versatile in application because we can use bioleaching to recover things from ore, from coal fly ash, even from electronic waste. And think about how much electronic waste we generate as a world. Also other waste processes. So as researchers are looking at this and learning and engineers are thinking about the design of facilities, I think we'll see bioleaching become a better part of what we're doing as a whole. So hopefully what I've done in the first few minutes of this presentation is to set the stage that microbes are truly as amazing as I've been claiming that they are, and how much they're involved in all these processes. So now we can talk about how do we understand these organisms in our systems. So I want to show you a couple of tools and talk through them. And I know some of you have probably used these in other applications, but I want to be sure everyone's on the same page. So I'm going to give you a quick summary of each, and then we'll move into some case studies. So let me start by saying sampling is easy. People always worry about collecting microbial samples, and there's been an enormous amount of work that's been done for decades on this. So we can extract DNA easily from liquid samples that we have in processes. We can do soil and solid samples. For corrosion, we do tons of scraping samples through the process. We can look at biofilms that are formed or corrosive properties that are present in those systems. And then we've actually moved to being able within the past year to do extractions in the field, which I think is huge for many industries and for remote locations or for radionucleotide situations. This can be a really valuable tool, and I'm going to show you a case study for that. So let me start by talking about something, if you've heard me talk before, I've said this word a lot, it's quantitative PCR. So qPCR is what we do now to count the microbes that we're interested in. So we can look at a microbe by name, or we can look at a group of microbes and what they do, like sulfate reduction or iron reduction. So when we tried to do this before with culture, as I mentioned, we'd have to take the sample, ship it to the lab, try to replicate the conditions, grow it on a plate, and it was just really unwieldy to be able to do it. With DNA-based technology now, we take the sample from the environment, extract the DNA from that sample, and then we use something called polymerase chain reaction, or PCR, to amplify an organism or a gene that we're interested in, and we can count how many of that we have in our original sample. And the pandemic really brought about the value of qPCR. We saw with COVID identification and quantification, it was a common household name. So now I don't have to describe this process as much as I used to. We've been using it in the medical industry and in environmental remediation literally for decades now, and it's a very valuable tool. But now we've proven time and again that this is the most accurate way to quantify different organisms and genes. And then the other thing that we can do, so now we know we have this valuable tool, and I can tell you how many sulfate reducers you have in your sample. But what if, like in mining, we need to look at a lot of processes? So I want to look at ammonia oxidizers, and I want to look at manganese oxidizers. We have a platform called QuantArray that you can see here. So this is a slide that has a square, and it has a lot of little holes. So then we can do qPCR reactions on a very small volume in each of these holes. So we generate quantitative numbers of each of those organisms or processes that we're interested in, so we can do it all at one time on a sample. So this gives us a lot of information that I'm going to show you. So there are a lot of different ones, and as Jonas mentioned, we do this a lot in countries all over the world, including Australia. So all the sampling supplies, everything is there that you guys need, and we can do it for bioremediation purposes, for chlorinateds or for petroleum hydrocarbons. Today I'm going to focus and show you a case study where we used it for corrosion in a mining system, and I'm also going to focus on an array that we call the BGC, or biogeochemical array, and this is a great tool for mining, but there are others available depending on the questions that we want to answer. So let's start talking about what I mean when I say biogeochemical. So the BGC literally stands for looking at biology, geology, and chemistry all together. So just like us, microbes need certain compounds to live and thrive and grow. And you've probably seen this if you've taken any micro classes or remediation classes in college, where you see the redox ladder. So a lot of organisms are aerobic, and they can do degradation very quickly or utilize oxygen to grow. And then when oxygen becomes depleted, you have other organisms that come in to use the nitrate, and we start to move down this ladder till we get to very reduced conditions when most things have been used at a site, and then you'll have more methanogenic. So in mining, we can have all of these that happen as there's changes in flow and movement through systems. So understanding this entire ladder is very important to us. So we developed this biogeochemical array with ExxonMobil a few years ago for use in remediation, but it has great application here too. So I'm showing you a really complex diagram, and I'm happy to share these slides if anyone wants to look at this more. But basically what I'm trying to get you to see here is there's a lot of interaction between these processes and between the organisms. So the blue boxes you see are chemical and geochemical metrics that you guys measure on a regular basis for environmental samples or for mining. What the yellow boxes are the organisms that use these compounds. So then you can see the arrows of how they transfer things between each groups of organisms as different compounds are changed as well. And so the yellow box gives us the bacteria, and when it has a red line around it, those are aerobic conditions. The black line are more reducing conditions as we move down that redox ladder. So this kind of helps to show you the complexity of the environment, and all of these yellow boxes are what we want to look at when we're thinking about biogeochemistry. So when do we use this? Well, if you want to characterize the genetic potential that's present, so I've got this certain metal, say gold, and I want to precipitate it better at certain parts of my process, which organisms do I need? We can start to see if the right redox conditions are present for that precipitation to occur. What if you have acid mine drainage and you're thinking about different treatment strategies, or you want to kind of mitigate it early in the process? We can use this to help us understand how the organisms are surviving under these more acidic conditions. And then finally, what if you've closed an area and it's your job to help with revegetation and restoration? We can use this particular array to also help us understand nitrogen cycling and nutrient cycling, so we can see how healthy the soil and the groundwater are in these different systems. So a lot of uses throughout the life cycle of a mine. So before I dive into the data, I just want to show you a little bit about how this looks. So there's 25 different genes that we look for in the QuantArray BGC, starting with higher level organisms like total bacteria, then moving through the redox ladder, looking at sulfur cycling, iron cycling, denitrification. So a lot of different processes, and you get for each sample a number that's associated with that particular group. And so what you can do then is you can look and go, wow, I'm really interested in iron reducing bacteria, and I can see that they're present here in this sample at about 10 to the 6 cells per gram. So what does that mean? Well, we also give you some help with the interpretation. So there's a heat map that you can see here, and it'll be shaded in the green to the red. So green is high concentration, red is low concentration. So we've analyzed around 250,000 samples from sites all over the world. And so I can go into the database and look at iron reducers and see how many we've detected at 10 to the 6. And then that lets us know from a percentile distribution of those 250,000 samples if that's a high abundance or if it's a low abundance. So in this case, I can go, wow, there's a lot of iron reducers, a lot of potential there. I've got some nitrogen cycling that's also happening at a pretty high degree, and some acid production. So it can start to tell me what's going on in this particular sample, and I can compare that across locations. So hopefully I've shown you qPCR is a very useful tool for us in mining. Another one that's useful, and I promise I'm only going through 2, so I'm not going to hopefully overwhelm you too much talking about molecular tools. But there's something called next generation sequencing that is very common and has a big benefit in mining. So with qPCR, we were going in and quantifying a certain gene that we were interested in. So we knew we want iron reducers. We know we want sulfur reducers. But what if we don't know which organisms in particular, or if we want to just look at the whole community? So sequencing gives us the ability to do that, where we go in and we amplify all the microbes that are present, and then we sort them by name. And we know how many of each name approximately we got, so we can do some relative abundance of that community. And then we can use this community profile to help give us some other information. So we want to use something like next generation sequencing if you're trying to understand the total community that's there. And then if you want to see how that community is changing over time or across a location. And also it's great to use it if we don't have qPCR targets available. Like what if this is a new process that the mining industry researchers have just figured out? We can go in and look, do we have the name of that particular organism that they identified using this type of analysis? So this is really important for us in acid mine drainage, because we can go in and look to see if we have that acidithiobacillus that I talked about earlier, which can precipitate the iron and copper and zinc and arsenic. Or do we have iron oxidizers or iron reducers that are present in our system? So we can go in and look for very specific organisms that we're interested in. And some of these, like arsenide oxidizers, we don't have assays for yet. So this can be a very beneficial tool for us to use. It's also really, really beneficial for us for rare earth element recovery. What we're learning is this bioleaching of certain rare earth elements is better with certain genera of organisms than others. So with some bacteria and some fungi, we can start to bioleach different elements that we're interested in, like a monzonite, or different light or heavy REEs. We can start to do better bioabsorption, bioaccumulation, and bioleaching by knowing what these microbes can do. So if we do a sequence and we see that we have a lot of this micrococcus that's present, that means we have a really high likelihood for bioabsorption of heavy REEs that's happening in this particular sample. The data looks like this, and I'll move into kind of sharing some case studies with you quickly after this, but there's different samples that are listed at the top. We know how many sequences that we get in each sample, and then we classify them to the name of the microorganism to a genus level identification. We can see how diverse they are. So if we have a very acidic environment, do we have a lot less diversity in that particular sample compared to more neutral location? So we can start to look at those things as well. And then you're going to get something that looks like this, which is a pie chart that you can see on the right. So you can go, okay, there's a lot of iron reducers, Geobacter, an iron reducer, about 18% of what we see in that sample comes from there. So I've probably got a good bit of iron reduction going on. There's also some other anaerobic organisms that can be using different compounds. So it'll give you ideas of what's happening in the process, and you can start to compare these with different samples to understand it better. Then we can do things like statistical analysis to help us really pull out those differences. So we can take each of those genera names. In some samples, you might get 600 different genera and maybe 600,000 sequences or 1 million sequences that are detected. So if we use statistical analysis like hierarchical clustering that you see here, we can take all the names of those organisms and we can see, wow, location 1 and 2 are pretty similar. 3, 4, and 6 are more similar to each other. 4 and 6 are the closest. So you can start to see where things are similar, where they're different. And then we can do like principal component analysis as well, where we take all those genera name and put it into this statistical analysis of PCOA. And we can take and look at a variety of samples to see how they group. So if you have 5 different sampling locations here, we might see that these group together in more of an impacted location. These have less impacts that are present in these samples, so they tend to group together more. And then if we look at them over a time series, so where you see -1, -2, 10-3, 10-4, these organisms, you can see the community is changing over time. So then we can start to see how stable is the community as things flow through that system over time. So we can really see good distribution with these tools and get a good overall assessment of what's happening in our systems. So as I wrap up with NGS, the best way to use this is to help us look at our community population over time in response to a change that we're making. And we can see who does best under certain situations. So are we stimulating organisms that can do bioabsorption and bioaccumulation in certain situations? And then are we stimulating more methane producers that we may not want in this particular situation? So this type of analysis can help really give us some good information on that. Okay, so let me show you a couple of case studies here. The first is a merge pond that was being used for the treatment of heavy metals. And the goal of this case study was to understand how the microbial processes occurring in this pond are either contributing or not contributing to heavy metal reduction. So we looked at two main samples, an inlet sample and an outlet sample from this pond. And they were hoping between those two samples that these heavy metals were precipitating out and we'd see microbes that were related to those processes so that we could help understand what's likely to precipitate and what won't. So we did the QuantArray BGC that I just introduced you to for the analysis on both of these. So if you remember, we have this big chart here. So the first thing I'm going to do is really dive into nitrogen cyclers at the top here. And this is a little pop-up here, shows you the nitrogen cycle. The good thing is researchers have identified the genes all the way around this cycle. So whether we're looking at revegetation or we're looking at what's happening with metal changes and absorptive capabilities, we can see everything from nitrogen fixation, nitrification, denitrification. So we can really target all parts of this cycle. So let's look at some of the genes and the data. So in this chart, the blue bar is our inlet and the orange bar is our outlet. And the first group that I'm going to zoom into there, AMO, are our ammonia oxidizing bacteria. And these organisms are typically pretty tolerant of high concentrations of metals. And although they don't directly precipitate the metal themselves as a primary production, they will influence the chemical environment that affects the mobility and the bioavailability of the metals. So in this chart, we can see that they start out pretty low, but when we get to the outlet, we have a higher concentration by about 2 orders of magnitude. So if you move to the next group of bars, NIF, these are our nitrogen fixing bacteria. And this group is very important as they can be easily inhibited by heavy metals. So we were worried about that because metals will actually inhibit the activity of the nitrogenase gene that they use for fixation. And here we see that not to be the case, right? We have a good concentration around 10 to the 4 at the inlet. And at the outlet, we have almost 10 to the 8 cells. So it doesn't seem to be a concern at all in this situation. And they're not only present, but it looks like they're thriving there at the outlet of this area. So nitrogen fixing bacteria are really important in this process because they can remove heavy metal ions from solution. They use biosorption, and they can also help us to precipitate these heavy metals in general. That's exactly what this holding pond was. Now let's look at that third and fourth set of bars together. NIRS and NIRK are genes that are needed for denitrification. So denitrification is important for a variety of reasons, and you can see at the outlet we have pretty good concentrations, 10 to the 7 and 10 to the 4. There are some species of denitrifiers that can precipitate metals like zinc and cadmium and nickel as carbonates and hydroxides. Others that can co-precipitate the metals with calcium carbonate or other minerals. So they can also help us control the pH that's being generated in our system and further precipitate different metals via this pH shift that occurs. So finally, let's look at the last group of bars, which are our nitrate oxidizing bacteria. This is a fascinating indirect connection. Some species couple the reduction of nitrate with the oxidation of ferrous iron. So this is going to generate ferric iron oxides, which most of us know are insoluble. So when this biogenic iron oxide can be a powerful adsorbent and lead to the co-precipitation of or absorption of heavy metals, metalloids, and even radionucleotides. So if that was a concern in this system, that would be a very important group of organisms for us to look at. And again, we see they're in very high abundance in our outlet sample. Okay, so let's move to another section of the array and focus on iron and manganese cyclers. So similar to nitrate oxidizers, our first group of bars here are iron oxidizers, FeOB, and these iron reducing or iron oxidizing bacteria can help with the precipitation of iron if that's a concern in our system. The second group are equally important. Iron reducers can directly precipitate metals by reducing the metal ion to their elemental form. For example, some IRB can lead to gold precipitation in a system. So if we had gold that we wanted to precipitate out, making sure we have enough IRB would be very important. And then others can help precipitate magnetite or siderite. And then finally, our last group of bars, MnOB, that you see are our manganese oxidizers. And as manganese is oxidized, it leads to the precipitation of manganese oxides, which also absorb and co-precipitate metals. Okay, last one, I promise, and then we'll move on to a different case. Look closely at our last two groups, our sulfate reducers and our fermenters. And this time we're going to think about these different groups kind of together as a whole. So the first set of bars are our APS gene that's found in sulfate reducing bacteria. And I've mentioned sulfate reducers quite a number of times because they're really important in a lot of systems. So as sulfate reducers produce sulfide, the sulfide reacts with dissolved metal ions such as iron, zinc, copper, and many others, and they'll form these metal sulfides. And since they're generally insoluble, that helps to precipitate out of solution. So again, really good sign for this holding pond, and you see we're at almost 10 to the 8. And then our last group of organisms, the FER, are fermenting bacteria. And these are a little different, right? We can see the concentrations of these are similar between the inlet and the outlet. And we know that fermentation plays hand in hand with many of the other groups we saw, creating the conditions that we want in this situation. So they're conducive to creating reductive processes that we've been looking at. And while they don't increase across the system, they're pretty consistent. So every marker that we looked at in this BGC array, and as you could saw, that was a lot of different markers that we just walked through, showed very promising data that the microbes are highly active in this system and helping to demonstrate that we have the genetic potential for this metal precipitation to happen in this pond. Okay, so one more case study, and then we'll do the end of this and then wrap up with the future of where we're going. So one of the case studies we had is for a uranium mine. And in this uranium mine, they were seeing corrosion that they were observing throughout the system. And they wanted to know if microbes were present that are involved in microbially influenced corrosion. So the problem we had is, since this is a uranium mine, radioactivity is really a concern. It makes shipping of the sample really difficult. It also makes where you have to find a lab that is RAD certified, so it can make receiving those samples really difficult as well. And in the past, it's meant that we haven't been able to accept those types of samples. But the good news is, if you remember, we've talked about new field extraction capabilities. So for the last 25 years, I have really wanted to find a way that we could extract samples in the field. Because when we extract, we're taking cells that when we remove from the environment, those cells are live. They're still changing, they're exposed to oxygen, they're exposed to new temperatures and new compounds that are now bioavailable to these cells, so they'll start to change. So we need to extract the DNA out of that cell as quickly as possible, because then DNA is very stable. And so sometimes if we're at a remote location, or we're in a location like this where we've got radionuclides, we can't easily do that because we can't handle them in the lab or get them quick enough in the lab to not have a problem. So for the last 25 years in my career, I've really been working on trying to develop a field extraction method that was easy to use, that didn't require equipment in the field, that provided us with good, high quality DNA that we could use for assessment. So we now, last year, started offering something called the BioExtract, which we can use in site locations. We're using this in many different countries around the world now, in remote locations. We also used it in this particular situation to help us overcome the radioactivity, because what we could do is, they had 5 metal coupons and they wanted to look at this cycle every 45 days for 4 cycles to see if corrosion was increasing and what we were seeing colonizing these coupons. Well, since we couldn't extract them in the lab, they were able to extract them in the field in about 45 minutes for all of these coupons and to get the DNA, so then we could analyze the QuantArray, and this time we did the QuantArray MIC for them. And so it was very exciting, and what they did was measured, they looked at the radioactive concern in the DNA after extraction, and I've seen this in working with universities around the world. Once we extract the DNA, we've removed that radioactive label that is there, and so we're able to then ship that sample and have pure DNA that we don't have to worry about. So it did work very effectively in this, and they were able to ship us the samples. And due to time today, and to be honest, just overloading you with too much information, I'm only going to show you one sample from one of the time points, just to give you an idea of what you could do. So with the MIC array, we get a same list like you saw before with quantitative numbers that are attached to all these groups of organisms. In this case, what we did was go in to look to see which ones were high. So you can see we have a lot of sulfate reducers, a lot of acid producers that are very high, some oxidation processes that are occurring on these coupons. So we have quite a few organisms in very high abundance at the top of detection that we're seeing in this particular corrosion coupon. So then we can start to correlate that as a multiple lines of evidence approach with the chemistry of the coupons and the pitting data from the coupons, and start to understand the likelihood for this to be microbiologically influenced, that it's occurring, and then they can figure out what treatment methods, whether it's a biocide or maybe different pigging schedules, that can be applied to really help to resolve this corrosion concern. I know that I've given you a lot of information, and before I wrap up, I just want to take just a moment before I get into the future of where we're going to give you some assurance. I know this is a lot of information, and I rattled off a whole bunch of genera names that most people aren't as geeky as me and don't love to hear them or try to say them or read those names. But I want to let you know you don't have to be a microbiologist to interpret this data or do this testing. When you get familiar with these results and which organisms are doing which processes, it really becomes very easy to interpret. Also, you're never alone. We are very niche in what we do, and with the labs that we partner with around the world, it's our goal to help clients through the process. So, whether you're talking to Jonas or Aliona, as we go through this, we are always happy to help you understand what this data means. So we can do everything from helping you choose where should you collect your samples to get the most value to answer your question. Where we do this all for no charge because we want to share our expertise in these areas. We also are happy to have a call with you to talk through your results when you get them, so we can share what the microbes are doing. And we are not consultants. We are not design engineers. We don't sell any products because we really focus on just being microbiologists. So we want to be that part of your team. And we have different levels of reports that are available to you guys. We can do the standard report, or we can add a more comprehensive interpretation about the microbes in your system if you would like for us to do that. Okay. So now I've gotten to the fun part. For those of you that have more expertise, especially, I think you're going to enjoy what is coming out here. And I can't share everything, but I would like to share with you at least a primer for what I think the future of mining holds, and I'd love to hear from you guys about what else you think is going to happen. So what are we doing as we move forward in the future? So I'm under an NDA that I can only disclose some of the publicly available information on this. I may not be able to answer all of your questions, but we can at least talk through kind of the general idea of what happened here. So these are slides. I'm going to give you a glimpse into the future of microbes and mining processes. And these are slides that were created by the Battelle Memorial Institute in the US. They were funded by the Defense Advanced Research Projects Agency, or DARPA, which is with the Department of Defense in the US. And the goal of this project was to help facilitate the separation of metals and metalloids, and in particularly focus on rare earth elements. So as I alluded to throughout the presentation today, a lot of mining processes, including that that we use for recovery of rare earth elements is expensive and it's very cumbersome. We have strong acids that are needed to leach the compounds for recovery. Then we have large quantities of organic solvents that are needed for separation. And not only that, you have to go through hundreds of purification stages to get to really good, clean separation, and then sometimes transform the compounds at the end. So it is a very costly process that takes a lot of time, generates a lot of waste. It can lead to failures that cause damage to ecosystems and even closures of mines that have happened and shutdowns due to problems in this system. So they began their journey to help identify proteins that bacteria naturally produce so we can harness the power of these to improve the recovery of REEs in particular through more cost-effective and less toxic processes. So a lot of the research that Patel was able to do resulted in finding a region of interest in several bacteria, and you can see it here on this slide. There are some calcium-dependent areas that are found in secreted proteins from bacteria, and they reversibly bind calcium at 8 different binding sites. So you can get a lot of binding capacity in this particular situation. So what they did was they added a fluorescent protein to the end of the site that you can see there. And then they observed the conformational change, which indicated that these rare earth elements were binding into this domain. And so then you would have excitation of this probe and release. And this was through a technology called FRET, which is fluorescence resonance energy transfer. And so using this tool, they were able to confirm that the binding happens and look at different rare earth elements of interest to be able to get those KD values. And then what they did was they used this technology to help them identify what stable calcium binding sites they wanted to look for. So then they went out into literature and they found all of these binding sites. And then they used something called an alpha fold modeling to help them predict and down select which ones were going to be the best to pursue. And they ended up coming out after looking through, literally thousands of these and came out with 5 candidate proteins that they wanted to use for further characterization. And so here are the REE binders that were from these 5 different proteins. And again, I can't get into all the details, and I am not an expert to be able to share all of what they did, but there's a great paper that came out in ChemSci in May of 2025. Corey and Heater, if you want to look it up, that's a great publication that can give you all the publicly available research that went behind identifying these candidate binders. So after a lot of study and investigation, the end result of where they are now is they've developed proteins that lead to the separation of complex mixtures of REEs. And they found a protein in particular, HU5, that ended up being the best performer. And the separation was performed at a pH of 3. And the isolated fractions contained REEs with purities ranging from 69 all the way up to 91%. So this work has been patented and it's now being tested. They did laboratory bench scale studies and they even went down to a pH of 1.5. And so this is particularly useful for us in mining operations. And they had column studies that were successfully performed for 2 years with continuous flow and they were still stable. So I think this gives us an indication that this can be moved to a larger scale system, and with engineering design, we can scale this up to be able to use in mass production, to be able to separate these in a very low cost, very efficient way with minimal toxic compounds that are produced using these naturally available proteins to help us recover and bind these REEs at really remarkable purity levels. And there's a lot more they can do. And they found that they can do not just REEs, but there's other metals of interest that can be purified, things from looking at improved purity of iron, and gold recovery, and lots of things that can be done as we learn more about these proteins and this separation. So there was a pretty massive team, as you can imagine, that was involved, and a lot of time and effort that was involved in this research. So a phenomenal team that really worked on this substantial development. And I really think it's the future of where we're going. And if I go back to this question, you know, are we going to end up installing bioreactors that have proteins in it that can help us bind at 95% efficiency for some of these compounds from our system? So if you could recover instead of 70%, 95% from a solution, I mean, think about how much cost benefit there is just in that alone. And if we can do it without hundreds of separation processes and toxic compounds, and we're using microbes and harnessing the proteins that they naturally produce that aren't harmful, I think what we're moving forward into in mining is a whole new world. And I obviously am not an engineer that designs these at sites or has the mining experience that a lot of you do, but I think if we work together as a community and we get this technology so that it's commercially available and can be tested at sites and optimized and refined, I think we're going to answer these questions that I have and a lot more. And so I look forward, because I think there's a lot of brilliant scientists in this industry, or that may even be on this webinar tonight. And so I hope we can work together to really change the future of mining and to embrace some of these new technologies, so we can start to think about what we can do together to not only have a very much improved cost benefit, but also to have less liability and less waste that we're having to deal with as these processes move forward. So I'm personally very excited about the future of where we're going in our industry, and I hope you guys feel that enthusiasm from me and can see the value of it, and that we can work together moving forward to try to bring this into reality for us.